Hello everyone, today we are from Group 3. We will present our work for course STB1033 General Genetic. We will dive into groundbreaking work of James Watson and Crick, who unveiled the DNA structure. Their discovery, built on research by Rosalind Franklin and Maurice Wilkins, revolutionized molecular biology. This breakthrough has shaped genetics, medicine, and biotech, stating for Group 3 insights into Watson and Crick's remarkable contribution. Thank you. Next, for our content, we will explore the significant breakthrough by Watson and Crick, understanding the simple beauty of the double helix. We will explain the history and structural aspect of DNA, including its encoding, base pairing, and anti-parallel orientation, showcase its crucial function as life blueprints.
History. In mid-20th century, James Watson and Francis Crick, drawing on decades of scientific research, unveiled the groundbreaking structure of DNA. Watson's background in zoology and exposure to Linus Pauling's work on protein structures, along with Crick's expertise in X-ray crystallography under William Lawrence Bragg, fueled their collaboration. Watson's fascination with DNA began after a 1951 conference where Maurice Wilkins presented X-ray photographs of DNA. Watson and Crick, driven by the molecular model successes of Pauling, rapidly constructed DNA models. The pivotal contribution came from Rosalind Franklin's unauthorized X-ray image. In 1953, they published the now widely accepted three-dimensional structure of DNA in Nature. The discovery of James Watson and Francis Crick. In 1953, they concluded that the DNA molecule appears as a three-dimensional double helix. Rosalind Franklin and Maurice Wilkins, they used X-ray crystallography to study DNA structure, which helped Watson and Crick with their discovery. Characteristics of DNA. DNA. It is a deoxyribonucleic acid that made of a nucleotide monomers. While the DNA double helix, it consists of two nucleotide chains. Each nucleotide consists of a sugar molecule, a phosphate group, and a nitrogen-containing base. Structure of DNA. Hydrogen bonds between complementary bases hold DNA's two polynucleotide chains. The base pairs are adenine A bonds with thymine T and cytosine C bonds with guanine G.
Did you know the sequence or order of the bases in a specific DNA strand is related to the expression of specific traits and qualities? For example, the sequence GTC-ATT might be responsible for curly brown hair, while GTC-AAT might be responsible for straight black hair. Important features of model. The Watson and Crick model of DNA is a double-stranded helix. Phosphate molecules make up the backbone of each strand, and the complementary bases bond along the rungs of the twisted ladder shape with hydrogen bonding. The two connected strands of DNA run in opposite directions, making them complementary to each other. Function of DNA. It stores genetic information necessary for organisms to develop, survive, and reproduce. Damage to DNA may cause cells to stop dividing, cell death, or become malignant. Anti-parallel orientation. Double-stranded DNA is an anti-parallel molecule, meaning that it's composed of two strands that run alongside each other but point in opposite direction. In a double-stranded DNA molecule, the 5N phosphate-bearing end of one strand aligns with the 3N hydroxyl-bearing end of its partner and vice versa. Summary. The discovery. The double helical model came from the combined work of Watson, Crick, Rosalind, and Wilkins. DNA characteristic. DNA is made of nucleotide monomers. The four bases are adenosine, thymine, cytosine, and guanine. DNA function. DNA stores an organism's genetic information.